Welcome to G Fun Facts Online. Imagine a cancer cell. Deep inside its nucleus, you'd expect to find our genetic blueprint, the DNA, neatly organized into 46 chromosomes. But in many of the most aggressive cancers, there's chaos. Tiny, untethered loops of DNA are running rampant, existing completely outside the normal chromosomal structure. These are called extrachromosomal DNA, or ecDNA. For decades, they were little more than a biological curiosity. Now, we know they are a primary reason why cancer is so aggressive, so adaptable, and so difficult to beat. This is the story of how these renegade DNA circles are rewriting our entire understanding of cancer. Our story begins not with a supercomputer, but with a simple microscope. Back in the 1960s, pathologists peering at tumor cells noticed something strange: tiny, paired dots of chromatin that were clearly distinct from the large chromosomes. They gave them the descriptive name double minutes. These observations were intriguing, but the technology of the time was limited, and for many years, double minutes were considered a rare aberration, a strange quirk found in only a few tumors. The tide began to turn with the rise of molecular biology. Scientists started to connect these double minutes to a process called gene amplification, where cancer cells make many extra copies of certain genes. This was a crucial insight, but the true scope of ecDNA's role remained hidden. The real paradigm shift didn't happen until the 2010s. With the convergence of high-throughput gene sequencing and powerful computational tools, research teams, like one led by Dr. Paul Michel, made a startling discovery. These DNA circles were not rare at all. In fact, they were present in a significant percentage of many cancer types, especially the most aggressive ones like glioblastoma, sarcoma, and esophageal carcinoma. The biological oddity was officially promoted to a central player in the narrative of cancer. So where do these rogue DNA circles come from? They are forged in genomic chaos. One of the most spectacular formation mechanisms is an event called chromothripsis. The name literally means chromosome shattering. In this catastrophic event, one or more chromosomes are pulverized into dozens or even hundreds of fragments. The cell's repair machinery then frantically tries to stitch these pieces back together, but it does so in a completely haphazard way. In this mad scramble, some of the DNA fragments can be ligated end to end, circularizing to form a new ecDNA molecule. Another violent pathway is known as the breakage-fusion bridge cycle. This destructive process often starts when a chromosome loses its telomere, which is the protective cap at its end. The newly exposed, sticky end is seen by the cell as a break and confused with its sister chromatid. During cell division, as the cell tries to pull the chromosomes apart, this fusion creates a bridge that gets stretched until it snaps. This breakage creates even more genomic instability and can release fragments that then circularize into ecDNA. It's a vicious cycle of damage and faulty repair that fuels the creation of these powerful cancer-driving elements. Once formed, ecDNA becomes a potent weapon in the cancer cell's arsenal. Its circular shape and its freedom from chromosomal constraints give it several deadly advantages. One of its most critical functions is the massive amplification of oncogenes. These are the genes that, when mutated or overexpressed, put the pedal to the metal on cancer growth. ecDNA is a major carrier of some of the most notorious oncogenes, like MYC, EGFR, and MDM2. While a normal chromosome might have one or two copies of a gene, a single cancer cell can contain hundreds of copies of an oncogene spread across numerous ecDNA molecules. Studies have consistently shown that this ecDNA-driven amplification is common across many deadly cancers and is almost always associated with a poorer prognosis for the patient. The high copy number leads to a flood of cancer-promoting proteins, giving the tumor cells a huge growth advantage. But the power of ecDNA goes beyond simply increasing the number of gene copies. The very structure of ecDNA makes the genes it carries hyperactive. In our chromosomes, DNA is tightly wound around proteins into a compact structure called chromatin. This packaging helps keep genes switched off until they are needed. ecDNA, however, has a much more open and accessible chromatin structure. This means the cellular machinery that reads genes and transcribes them into RNA has a much easier time accessing them. The result is a level of gene expression that is significantly higher than what you'd expect from the copy number alone. It's not just that there are more copies of the gene, each copy is working in overdrive. Furthermore, ecDNA can act as a mobile enhancer. Enhancers are short regions of DNA that act like volume knobs for genes, boosting their expression, sometimes from a great distance. When an enhancer is located on an ecDNA molecule, it can powerfully boost the expression of an oncogene on the same circle. This creates a self-contained, high-powered system for oncogene overexpression. Recent discoveries have revealed another layer of ecDNA's power, its ability to form hubs. These are clusters of many different ecDNA molecules that congregate together in the nucleus. Think of it like a specialized workshop where all the tools and workers for a specific malicious task are brought together for maximum efficiency. Inside these hubs, enhancers on one ecDNA molecule can activate oncogenes on a completely different ecDNA molecule. This allows for a coordinated and massively amplified assault on the cell's normal regulatory systems. Scientists have even identified a key protein, called BRD4, that acts as a kind of molecular glue, holding these hubs together. This discovery, as well C, provides a tantalizing target for future therapies. One of the greatest challenges in treating cancer is its remarkable ability to evolve and adapt. A tumor is not a uniform collection of identical cells. It's a mosaic of different cell populations, a phenomenon known as intratumoral heterogeneity. ecDNA is a primary engine driving this diversity. When a healthy cell divides, it uses a structure called a centromere on each chromosome to ensure that the duplicated chromosomes are precisely and equally segregated into the two new daughter cells. ecDNA lacks a centromere. This means its inheritance is completely random. When a cancer cell with 100 ecDNA molecules divides, one daughter cell might randomly get 70 of them, while the other gets 30. Or, one could get 99, and the other just 1. This process, known as non-Mendelian inheritance, rapidly generates a huge amount of genetic diversity within the tumor. Cells that happen to inherit a higher copy number of ecDNA carrying powerful oncogenes will have a growth advantage and will be selected for, allowing the tumor to evolve and become more aggressive over time. This is Darwinian evolution on hyperspeed, all happening inside a single patient. This dynamic and heterogeneous nature makes ecDNA a formidable adversary when it comes to cancer treatment, as it plays a significant role in developing resistance to therapy. Consider targeted therapies, which are modern drugs designed to specifically attack the products of cancer-causing oncogenes. Let's take the example of glioblastoma, a highly aggressive brain cancer where the EGFR oncogene is often amplified on ecDNA. When a patient is treated with an EGFR inhibitor, the drug attacks the cells that depend on that oncogene, but ecDNA gives the cancer cells a clever escape route. Some cells can simply discard the ecDNA carrying the EGFR gene. Without the target, the drug becomes useless against them. These cells can then lie dormant. The moment the treatment pressure is removed, the ecDNA can reappear and the tumor can come roaring back. It's a terrifying biological game of hide and seek that makes lasting cures incredibly difficult. This isn't limited to targeted therapies. ecDNA also drives resistance to conventional chemotherapy. For instance, ecDNA can amplify genes that code for drug efflux pumps. These are proteins that sit on the cell surface and actively pump chemotherapy drugs out of the cell before they can do any damage. Ironically, some chemotherapy drugs, which work by causing DNA damage, may inadvertently promote the very chaotic events, like chromothripsis, that lead to the formation of more ecDNA, potentially fueling a vicious cycle of treatment and resistance. The growing understanding of ecDNA's central role in cancer has, however, opened up exciting new avenues for diagnosis and treatment. Researchers are now actively developing strategies to detect and target these renegade DNA circles. The ability to detect ecDNA is crucial for diagnosis and for predicting a patient's prognosis. Advanced imaging methods like fluorescence in situ hybridization, or FISH, can be used to literally see and count ecDNA inside tumor cells. At the same time, advanced whole genome sequencing combined with specialized computational algorithms can identify and fully characterize the ecDNA from a tumor sample. But perhaps the most exciting area of research is the development of liquid biopsies. The theory is that as tumor cells die, they shed their ecDNA into the bloodstream. Detecting these circulating ecDNA molecules from a simple blood draw could provide a powerful, non-invasive tool. We could potentially use it to diagnose cancer, monitor how well a treatment is working in real time, and detect the emergence of drug-resistant ecDNA populations long before a relapse becomes visible on a CT scan. This could revolutionize how we manage cancer patients. Even more promising is the new frontier of therapies that directly target ecDNA's unique biology. Instead of just targeting the oncogenes that ecDNA carries, scientists are looking for ways to attack ecDNA itself. Several strategies are being explored. One is to inhibit ecDNA formation. Since ecDNA is born from DNA damage and faulty repair, drugs that block key proteins in these repair pathways are being investigated as a way to prevent ecDNA from being created in the first place. Another clever strategy is to exploit a vulnerability. The rapid, uncontrolled replication of ecDNA puts immense stress on the cancer cell's replication machinery. This creates a state of replication stress that makes these cells highly sensitive to drugs that target the cell's stress response. An experimental drug called a CHK1 inhibitor, for example, has shown great promise in killing cancer cells that are loaded with ecDNA. We are also developing drugs to break up those ecDNA hubs. By using inhibitors of the BRD4 protein, the glue that holds the hubs together, researchers can disperse the clusters and dramatically reduce oncogene expression. And finally, the ultimate goal is to find drugs that can selectively trigger the elimination of ecDNA from cancer cells, effectively disarming them of their most powerful weapon. The discovery of the hidden world of extrachromosomal DNA has fundamentally changed our view of cancer. These renegade circles of DNA are no longer a footnote. They are central architects of tumor evolution, aggression, and therapeutic resistance. The journey from the first observation of double minutes to the development of ecDNA targeting drugs is a testament to the power of scientific innovation. As we continue to unravel the complexities of ecDNA, we move closer to a future where we can outsmart even the most cunning of cancers, turning the tide in the fight against this devastating disease.